Today I invite Dr. Kahlilia Blanco, who is a scientist in our department, to give this grand rounds presentation. Dr. Blanco obtained her PhD from the Department of Neurology at the University of Miami, and she recently also obtained a career development grant from the American Heart Association, which is a fellow to faculty transition award, which starts today. Her grant starts today. And then Kahlilia is a very well accomplished and well-rounded stroke basic scientist. So to her credit, the Stroke, which is the flagship journal of the American Stroke Association, invited her to join the editorial board. And I heard that she is the youngest editor of the Stroke, which is a huge distinction. So Kahlilia is going to present her work on the role of epigenetics in post-stroke brain damage. Kahlilia, go ahead whenever you want.
All right. Are you able to see the screen?
Yes, Kahlilia.
Okay, great. All right, I'll go ahead and get started. So good morning, everybody, and thank you, Raghu, for the very nice introduction. Also, thank you to Raghu and Dr. Brooks for this invitation to present some of my research here at Grand Rounds. And as Raghu indicated, today the focus of my talk will be on the role of epigenetics in preventing brain damage and promoting recovery after stroke. And in particular, I'll be talking about how epigenetics can be used as a therapeutic tool in stroke research. So first of all, what is epigenetics? So epi means above or on top of. So from a very basic definition, epigenetics are structures on top of genes. And at the molecular level, epigenetics are chemical modifications of chromosomal DNA that can change the pattern of gene expression without altering the DNA sequence. And so this occurs through the addition or removal of biochemical structures located on the DNA, so that the presence or absence of these biochemical structures can cause a gene to become active or a gene to become silent. So basically to turn a gene on or to turn a gene off. Again, on the cell and molecular level, these epigenetic changes occur on the DNA, and this can be something like DNA methylation, which is the most common DNA modification, or these modifications can occur on the histones, which are the proteins that wrap and organize DNA into chromatin, and these modifications include things like acetylation, phosphorylation, and methylation. So the fact that epigenetics can turn genes on and turn genes off means that epigenetics can be just as powerful as genetics. And we know with genetics that there are variations in DNA sequences that can lead to changes in gene regulation, which is what establishes phenotype. And we know with epigenetics, which are modifiable by things like lifestyle factors or medical interventions, that these epigenetic modifications can also cause changes to gene regulation and regulate phenotype. And we also know that epigenetics is what allows each cell in the human body that all contains the same DNA to specialize and carry out distinctive roles. And so the impact of epigenetics on phenotype can be demonstrated very simply and very clearly from twin studies. And so for well over a century, researchers have compared characteristics in twins in an effort to determine the extent to which certain traits are inherited and which traits are learned from the environment. So eye color would be an inherited trait versus language, which might be an environmental trait. However, these twin studies are still relevant today and a very good example of the influence of epigenetics. So in this particular example, we're looking at chromosome 17, and one twin's DNA is labeled with a green label for its methylated DNA, and the other twin's DNA is labeled with a red fluorescent marker for its methylated DNA. The chromosomal photos are then overlaid, and then yellow indicates identical methylation patterns. And so we see in 3-year-old twins, you see lots of yellow. By the time these twins are 50 years old, you can see that there are significant differences. You see lots of green and lots of red, which shows how epigenetics can change over time in these genetically identical individuals. And so we know that disease patterns are not always the same in identical twins, despite having the same genetic mutation. And so that is just a powerful example of the impact that epigenetic changes can have on disease. So with the completion of the Human Genome Project 20 years ago, coupled with the explosion in the development of gene mapping, sequencing, and diagnostic tools, that has increased the ability of epigenetic gene regulation to be studied for the development of potential therapies. And so there's a large amount of research that are looking at how to combat diseases with what we term as epigenetic therapy. So epigenetics plays an important role in the brain, so much so that there is a subfield of epigenetics known as neuroepigenetics. And this is for three major reasons. First of all, several epigenetic marks are highly enriched in the central nervous system. So they occur at higher levels in the brain versus other areas of the body. And we also know that epigenetics play critical roles in the nervous system, not only in the development of the nervous system, but also in the maintenance of the nervous system. And this has been demonstrated not only in human postmortem studies or through animal models, but also in live human studies. So in this example, 5 years ago, a group of researchers actually developed and applied an imaging probe for an epigenetic enzyme known as HDAC. And they were able to visualize the expression of this epigenetic modulator in the living human brain through PET imaging. And so with this radio tracer, they're able to study region-specific distributions, and they're also able to profile differences in expression in varying developmental and disease paradigms. And so here is another example of how even within the live human brain, we can see how epigenetics might be playing critical roles within the nervous system. And so the third reason for the field of neuroepigenetics is that aberrant epigenetic regulation has been shown to contribute to the pathogenesis of various brain disorders, such as neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's disease within the aging brain, and then also within acute brain injuries, such as traumatic brain injury, or what I'll be discussing today, which is stroke. And so because of that, the modulation of the neuroepigenetic mechanisms may have some therapeutic potential because we're seeing that they're playing major roles in different diseases of the brain. So the pathophysiological mechanisms involved in stroke injury are complex and have limited the development of effective clinical therapies for treatment. For example, during ischemia, the deprivation of oxygen and glucose causes ATP levels to fall, causing loss of ion pump function, leading to degradation of ionic gradients and excessive release of glutamate. The excessive glutamate release causes high levels of intracellular calcium, activates damaging enzymes, and also overwhelms mitochondrial buffering capacity, all of which contribute to excitotoxicity. During the reperfusion event, the reintroduction of oxygen causes the production of reactive oxygen species due to abnormal enzymatic as well as mitochondrial function. And furthermore, the return of blood supply introduces inflammatory factors that cause disruption of the blood-brain barrier and lead to edema, which further causes injury. All of these mechanisms contribute to both apoptotic as well as necrotic cell death, which collectively cause brain damage following an ischemia reperfusion event. And so we see that there are a number of complex processes that are involved in ischemic stroke pathophysiology. And so mounting evidence over the last decade or so has revealed that epigenetics can actually regulate global changes in gene expression after stroke and therefore significantly impact functional stroke outcome. So after a stroke, altered epigenetic modifications cause a high balance of detrimental genes to be expressed, which leads to cell death. And so our goal is to modulate these epigenetic modifications with the aim of balancing the expression of survival or cell death genes, or better yet, even tipping the scales in favor of survival genes in order to protect the brain after stroke. So one good example of this is with DNA methylation, which is one of the most understood epigenetic mechanisms. And this is a very generalized description, but when methylation occurs in the promoter region of genes, it causes transcription to be switched off or causes gene repression. And so this occurs when enzymes known as DNMTs or DNA methyltransferases transfer a methyl group to the cytosine of DNA, and that causes methylated DNA. This methylation causes suppression of gene expression, and studies show that this methylation is involved in exacerbating stroke injury. So when you have global methylation or suppression of genes, this actually increases injury after stroke. And so in animal models, we see that mice with lower levels of this DNMT enzyme are protected from cerebral ischemia, whereas if you inhibit this enzyme with a drug, you can have improved recovery after cerebral ischemia. And then in human biomarker studies, we see that individuals that have 5-MC, which is DNA methylation markers within the blood, that this DNA methylation can be associated with ischemic stroke occurrence. And other studies have shown that levels of DNA methylation in the blood is a better predictor of ischemic stroke outcome than even chronological age. And so that kind of shows the power of epigenetics within stroke injury. So in addition to DNA methylation, which is a modification that has been studied for several decades, it was first identified in the 1940s or 1950s and has been studied in stroke for the last couple decades or so. A more novel epigenetic modification known as hydroxymethylation has been identified, and it seems to have the opposite effect to DNA methylation. And that hydroxymethylation, when it occurs in this promoter region of genes, is involved in turning on transcription. So it leads to gene induction. So opposite to what we see with DNA methylation. And so what we know about DNA hydroxymethylation so far is that it's most highly expressed in the central nervous system in comparison to other organisms or organs, and it's found at its highest levels in neurons versus other cells in the brain. And it has been shown to play important roles in neurodevelopment, synaptic plasticity, DNA repair, and other cellular functions. And so with DNA hydroxymethylation, this occurs when enzymes known as Depts produce a hydroxyl group onto methylated DNA to lead to hydroxymethylated DNA or DNA hydroxymethylation, which I will refer to as 5-HMC. And so what we know with methylation is that it suppresses genes and exacerbates stroke injury. And so with hydroxymethylation or 5-HMC, we know that it activates genes, but our question is, what role might it play in stroke injury? So to determine whether the epigenetic modification 5-HMC can protect the brain after stroke, we asked three major questions. Number one, is 5-HMC altered after stroke? Number two, does 5-HMC have an effect on gene expression and cellular function in the post-stroke brain? And number three, does modulation of 5-HMC contribute to neuroprotection or brain damage following stroke? So to answer the first question as to whether 5-HMC is altered after stroke, we obtained human brain sections from patients with ischemic stroke where a biopsy had been performed for diagnostic purposes. And the tissue was taken from the ischemic penumbra, which is the area that is adjacent to the dead necrotic core. And what we observed is that there was increased 5-HMC in stroke patients. And so here we were able to label the 5-HMC in DNA with a fluorescent green marker, and we see that that fluorescence increases in these stroke patients. We also co-labeled this tissue with NUN, which is a marker for neuronal cells, and we see that there is colocalization between the increased 5-HMC with this neuronal marker, which shows that 5-HMC is increasing in neurons in the human brain after stroke. So then we wanted to assess whether this phenomenon could be mimicked in an experimental animal model of stroke for further testing. And so for our model of focal ischemic stroke, we performed transient middle cerebral artery occlusion. Then we perform reperfusion, which allows the restoration of blood flow. And then we assess this peri-infarct region, again, which is the region that is adjacent to the necrotic core. And this region has the potential to survive if given some appropriate intervention. And so that is the region of the brain that we are very much interested in for therapeutic interventions. And so we repeated the study in mice, and we found the same result, that when we compared our sham controls to our MCAO, which is our experimental stroke model, we see that 5-HMC is increased and that this increase is occurring in neuronal cells. And to characterize this further, we also assessed TET activity. And so TETs, again, are those enzymes that are involved in producing 5-HMC. And we found that from as early as 5 minutes after stroke and then up to 1 day after stroke, that there was a significant increase in the activity of TET enzymes. And then when we isolated DNA and we used a probe against 5-HMC to test how much 5-HMC was in the DNA, we see from 5 minutes and up to 24 hours after stroke that there is a significant and sustained increase in 5-HMC. And so we see that this increase in 5-HMC seems to go up at around 24 hours where it peaks. And then starting at about 3 days, that increase in 5-HMC goes down. And by 7 days, the 5-HMC levels are similar to what we find in sham controls. So then we wanted to determine which TET isoform is involved in mediating this increase in 5-HMC. In mammals, there are 3 TET isoforms, TET1, TET2, and TET3. And so we ran a gene expression analysis and we looked from 6 hours to 24 hours after stroke. And we found that the TET3 isoform was significantly increased and that this increase was occurring in neuronal cells of the peri-infarct region. So to determine whether this specific isoform of TET3 is responsible for the increased 5-HMC after stroke, we used siRNA against TET3 to knock it down and then induce stroke. And we were able to confirm through mRNA analysis and protein analysis that led to a decreased levels of TET3. And then when we looked at 5-HMC, we saw that inhibiting TET3 led to loss of 5-HMC after stroke. And so this indicated that TET3 is responsible for the increased 5-HMC that we were observing after stroke. And so we were able then to answer our first question with these experiments, that 5-HMC is altered after stroke and that it increases. And so our second question was, does 5-HMC have an effect on gene expression and cellular function in the post-stroke brain? And so we performed a sequencing experiment where we could look at 5-HMC levels across the genome. And so what we observed, and here is our sham looking at this light purple line, is that we see low levels of 5-HMC in our sham controls. And then with MCAO, we see that there is a significant increase in 5-HMC levels across the genome. With the inhibition of TET3, we see that there's a loss of 5-HMC across the genome and that those levels go down, similar to what we see with our sham controls. And so we see this with our 5-HMC peaks, that it goes up with stroke, and when TET3 is inhibited, we have a loss of 5-HMC. And so then when we looked at the differential levels of 5-HMC in promoter regions, we see that with TET3 inhibition, it leads to a downregulation of 5-HMC in thousands of genes that are involved in a number of different processes within the cell, such as regulation of signaling, developmental processes, etc. And so we performed a gene ontological analysis in order to make sense of this data. And so this looked at various biological processes, and we found that TET3 inhibition led to loss of 5-HMC in genes that are associated with neuroprotection. So genes that regulate BDNF, which is an important neurotrophic factor within the brain, genes that regulate DNA repair or negative regulation of cell death, genes that are important for metabolism, such as response to oxygen levels or energy homeostasis, all of which are important after a stroke. And then we also found that there was a loss of 5-HMC in genes that are involved in neovascularization, such as angiogenesis and endothelial cell differentiation, etc. And so this indicated that 5-HMC could also be involved in promoting recovery after stroke as well. And so when we pulled out some of these genes, this is just 18 of the genes that have known function in ischemic neuroprotection. We then wanted to see whether the changes in the 5-HMC led to changes in gene expression. And so we ran a gene expression analysis, and we found that in 75% of the genes tested at the time point that we tested, that there was a significant decrease in gene expression that was associated with a decrease in 5-HMC. And so collectively, this answered our second question, that 5-HMC is associated not only with the expression of genes after stroke, but it is associated with the expression of neuroprotective genes after stroke. And so then thirdly, our most important question was, does the modulation of 5-HMC contribute to neuroprotection or brain damage following stroke? And so we used our same model of looking at Dept inhibition for loss of 5-HMC. And we found that with loss of 5-HMC, when we look at brain damage, we see that it exacerbates secondary brain damage after stroke. And so here with TTC staining that stains live cells red and dead cells white, and then looking at this these brain sections, we can see that when we compare our loss of 5-HMC with Dept inhibition to the controls, there is a significant increase in the amount of dead tissue or white tissue. And we found this for both the male as well as the female mice. And interestingly, when we looked at survival at, this is just 1 day after stroke, we see in male mice that almost 50% of the mice died with Dept inhibition. And so we did not see that same effect with the female mice. And so it kind of showed that perhaps male mice have a bigger dependency on 5-HMC for protection or for normal function than female mice do. So then we wanted to see if there were any impairments in motor function. And so we performed something called a rotarod test, which basically you put rodents on a rotating beam, and we can assess the amount of time that the rodent can stay on the rotating beam. And so with this test, with normal animals, they can stay on the rotating beam very well. However, animals that have had a stroke obviously cannot stay on the rotating beam as long. And so it can be just a very simple but a very good test to assess motor function impairments after stroke. And so here you can see how this works. We put the animals here, and these are healthy animals, so they are able to stay on the rotating beam very well. And so we actually did this experiment. These are rats, but we did this experiment in mice, and we see that loss of 5-HMC leads to less ability to stay on the rotarod. And so we have more motor function impairments with loss of 5-HMC. And so another motor function test that we performed was the beam walk test, which we just put a rodent on a tapered beam, and healthy animals are able to cross the beam fairly well, but animals that have had a stroke will have paw slips, and so we can actually count the number of times their foot slips, and that can be an indicator of motor function impairment. And so here, this is looking at rats. You can see this healthy rat is able to cross very easily. And in our experiments, we were actually using mice, so that the beam is a lot smaller, but we were able to quantify those foot faults, and we see that loss of 5-HMC led to a significant increase in the number of foot faults. Again, another indicator that loss of 5-HMC exacerbated motor function impairments. So with that simple experiment, we were able to see that modulation of 5-HMC can contribute to neuroprotection or brain damage following stroke. And it seems that 5-HMC provides a type of endogenous protection against stroke injury. And so then the bigger question for us then is, can increases in 5-HMC provide therapeutic protection against stroke injury? And so how can we modulate 5-HMC in a way that is therapeutically relevant? And the answer to that question, surprisingly, came through vitamin C. So vitamin C has recently been identified as an epigenetic regulator in addition to its many other functions. As you're well aware, vitamin C plays an important role in immune function, collagen synthesis, iron absorption. It's a powerful antioxidant. It's involved in regulating neurotransmission by regulating catecholamine biosynthesis. And now we also know that it's an epigenetic regulator. And so vitamin C can actually act as a cofactor for the Dept enzymes to drive their activity and lead to enhanced production of 5-HMC. And so vitamin C travels from the intestines, blood, or plasma into the cerebral spinal fluid, where it accumulates at high levels within the brain. And so thus for us, this seems like vitamin C may serve potentially as a good candidate for neuroepigenetic modulation because it's found at such high levels within the brain. And so we did some initial testing just to confirm that ascorbate treatment after stroke does in fact increase TET activity, as well as increase 5-HMC in the peri-infarct region after stroke. And in addition to that, we found that ascorbate treatment increased 5-HMC in neuronal cells of the peri-infarct region after stroke. And so then our kind of final question then was, can we test ascorbate treatment as a potential stroke therapy? So the Stroke Translational and Industry Roundtable, also known as STARE, is a consortium of medical doctors and researchers that was created to address the failure of neuroprotection by many drugs that have worked in preclinical animal models but have failed in clinical trials. And so this consortium called STARE suggests a number of criteria for preclinical therapeutic testing. So mode of delivery, therapeutic time window, testing in both sexes, looking in both young and aged animals, as well as stroke comorbid conditions. A lot of stroke research is done in young male mice, for example, and that may not kind of translate to something that's therapeutically relevant. And then also finding a drug with multiple mechanisms or targets. Because as I indicated to you previously, after stroke, there are a number of ischemia pathophysiological processes that are occurring. And so if you have a drug that only targets excitotoxicity, but then you have inflammation and oxidative stress happening, then it may not be as effective. And so finding something with multiple mechanisms or targets is preferable. And so we first wanted to just test ascorbate as a potential stroke therapy by looking at mode of delivery. We injected ascorbate IP intraperitoneal 5 minutes after stroke, so pretty much immediately after stroke, and then looked 24 hours later to see what would happen with brain damage. And we assessed a few different concentrations of ascorbate, and we found that ascorbate treatment at a 500 milligram per kilogram dose led to significant decrease in brain damage. In addition to that, we didn't see any increased levels of animal death or anything with these high levels of ascorbate. So also we assess regional cerebral blood flow, because we know that altered regional cerebral blood flow can modulate ischemic brain damage. And so here we just confirmed that the neuroprotection that is afforded by ascorbate and its abrogation by TET3 knockdown is not due to altered cerebral blood flow. So here we compared the saline groups to the ascorbate group, to a group with ascorbate and the TET3 siRNA knockdown, and we see that there was no alterations in regional cerebral blood flow. And then we also just tested this mode of delivery in both sexes as well as young and aged mice, and we see that in young and aged males and young and aged females that ascorbate treatment significantly reduced brain damage in all 4 of these groups. And in particular, in the aged males, the ascorbate treatment seemed to be most protective. So injecting something immediately after stroke at 5 minutes after stroke is not very therapeutically relevant. So we also looked at delayed ascorbate treatment. And so we injected ascorbate at 30 minutes, 2 hours, and 6 hours after stroke, and then assessed both brain damage as well as functional recovery at 7 days after. And what we found was that injection of ascorbate at 30 minutes, 2 hours, or 6 hours all significantly reduced brain damage. And also when we compare here, looking at injection at 30 minutes versus 6 hours, we see that there is the promotion of motor function recovery in both the 30 minutes group and the 6 hours group. However, the 6 hours group was less effective in that the promotion of motor function recovery did not always extend up to 7 days like we saw with the 30-minute group. So giving it at 30 minutes was more effective than giving it at 6 hours, but it was still very interesting to us that even giving it at 6 hours still modulated protection. So then we went ahead and tested this 30-minute time point of injecting ascorbate in both sexes, and we saw that there were improvements in motor function for both the rotor rod as well as the beam walk. the female mice seemed to perform better than male mice on these experiments. And then we also performed cognitive testing using the Morris water maze, where mice are placed in a pool and they're forced to try to find a hidden platform. And so we can assess both spatial learning as well as memory. And we found that both male and female mice that had ascorbate performed better on these tests, although female mice seemed to perform better than the males overall with the ascorbate treatment. And so then we also found that there was a reduction in lesion volume for both the male and the female mice. When we look 28 days later at 28 days of reperfusion, we see that ascorbate treatment reduces lesion volume, and that this reduction in lesion volume is abrogated by TET3. And so this is indicated to us that the protection that we were seeing could be occurring through the ability of ascorbate to modulate 5-HMC. And so finally, we wanted to look to see whether ascorbate is effective in both aged mice as well as comorbid conditions. And we found that in both aged male and female mice that there were reductions in brain damage and also improvements in motor function on both the rotarod as well as the beam walk. And we performed the same experiment in diabetic mice. Here we see reductions in brain damage. We performed a different motor function test because the diabetic mice are also severely obese. So they're not able to stay on the rotating beam, the rotor rod, and they're not able to cross the beam walk very easily. So we weren't really able to get any baseline readings. And so we used a different method to test motor function, which is an adhesive removal test, which is placed on the contralateral paw. And we see how much time it takes for the animal to remove the sticker off of that paw. And we see that for these diabetic mice that received the ascorbic treatment, they performed better on this motor function test. And so then we also tested hypertensive mice. And again, we saw a reduction in brain damage for both male and female mice, and also improvements on Neuroscore. And so finally, we also performed sequencing just to assess whether some of the protection that we're seeing via ascorbate was actually occurring through 5-HMC. And so when we performed our sequencing analysis and we look across the genome, we see that with ascorbate treatment, that there are significant increases in hyperhydroxymethylation that occurs across the genome, and that the 5-HMC is being regulated in a number of genes that are involved in ischemic neuroprotection, such as blood-brain barrier integrity, oxidative stress, and inflammation in particular seemed to be a process that was highly regulated by ascorbate in terms of having lots of 5-HMC in inflammatory related genes. And so finally, we were able to show that vitamin C is an epigenetic regulator with therapeutic potential against stroke. We know that vitamin C is a drug with multiple mechanisms or targets, and we found that vitamin C treatment, high doses of vitamin C treatment after stroke can be effective in both sexes, young and aged animals in stroke comorbid conditions, and that there is a good therapeutic time window and an easy mode of delivery for ascorbate treatment. So I would like to thank everybody in my lab for all of the help on all of the experiments that I presented today. I would like to thank Raghu, who is an excellent mentor. Thank him for his leadership, his mentorship, his patience at times, and his support of my research and my career, and also just for fostering a lab environment that makes it easy to be very passionate about research. And so I also like to thank all of our funding from the National Institute of Health, American Heart Association, Veterans Affairs, as well as the generous support of the Neurosurgery Department to the Vemaganti Lab. And so if there are any questions, I will go ahead and take those now.
Thank you, Dr. Blanco. That's an outstanding talk. And it's, we grew up with our parents telling us to have the balanced diet with vitamins. Now you're beginning to show some of the really remarkable efficacy. Well done. Other questions for Dr. Blanco, please? And Raghu, I'll let you be in charge of the question period.
Yes, Dr. C. Yeah. Go ahead, guys, if anyone has a question.
Kahlilia, this is Benny Iskandar. Thank you for this great talk. Does this give you a hint on the mechanism of diabetes-induced stroke or cardiac disease?
Are you saying is epigenetics involved in diabetes-induced stroke or cardiac disease? Is that your question?
I'm sure it is. The question is, does it give you a hint on the mechanism or the pathophysiology of how that happens?
Yeah, so I think it doesn't necessarily tell us the pathophysiology of how that happens, but I think that it definitely tells us that by regulating epigenetics, at least the 5-HMC modification, that can have a significant impact. And again, we're regulating something in a very global sense, as we see that across the genome, we see huge changes in 5-HMC. And so there's a lot of changes in a lot of different genes. And so I think it would take some additional research to try to kind of tease out exactly what's happening. But I think it would be interesting to kind of delve a little bit deeper and see exactly what's going on there.
Yeah, this would be an interesting, follow-up on the work.
Yes.
Hi, Kahlilia. This is Reed. Very nice talk and very nice project. Thank you for sharing it with us. I was wondering if you could comment a little bit more about the differences that you saw regarding the males and the females. And why do you think you're seeing such differences between those?
Right. So I didn't have time to kind of show all of the data, but one of the things that we found is that the induction of TET activity as well as 5-HMC occurs earlier in the female mice than it does in the male mice. And so with that, I think that's why we see that increased protection in the female mice versus the male mice. So that's kind of one explanation as to why we might see those sex differences. The second difference is that at least in the young adult female mice, those mice have smaller infarcts after stroke. And so a lot of times it is easier to get protection in the young adult female mice than male mice, just because the infarct size is a lot smaller. And so it's kind of easier to protect those. So there's kind of both of those things could be involved in those sex differences that we saw.
Okay,
Yeah,
great. Thank you.
and also to add a small point, even in older females, Kahlilia saw protection with ascorbate, and I feel that female brain is more plastic than the male brain, even at a younger age or an older age, and therapeutics work. Some of the therapeutics at least work a little easier in females than males.
Interesting. Okay, thank you.
Yeah, thanks everyone.
Well,
If
I had a
there
quick
are,
question too,
yeah, go
if
ahead, Shari,
that's
go
okay.
ahead. Yeah.
It's a really interesting talk. I was just curious, how are you guys defining the penumbra and the mice? And like, did you guys see any I guess differences in expression and like the 5-HMC or the TET expression in the penumbra itself as compared to like the core? Like what makes the penumbra and like potentially salvageable versus like the core? Did you guys see any differences?
Yeah, that's a great question. So we are specifically interested in the penumbra or the peri-infarct region because after a stroke, the cell death spreads. And so without therapeutic intervention, that peri-infarct region or penumbra, those cells will die. And so we are interested in that region because those cells are kind of susceptible to some type of therapeutic intervention. And so we're less interested in the core because those cells are already dead. And so I didn't actually go and look at 5-HMC changes within the core, although I do think, kind of to your point, it would be interesting to kind of look at more time points, maybe from early to late, to the area that would be the core and just see how 5-HMC levels change within that, what turns into that core region. So I think it would be really interesting to see just kind of how those epigenetic changes kind of change over time with stroke. But we really only looked in the peri-infarct region, which is the region that is just adjacent to the ischemic core.
Thank you.
Hi, this is Mahua. I have one question. Along the line what Reed and Raghu was saying, did you look at like estrogen and progesterone are known to have like protective effect for stroke? And astrocytes, it's not just the ovaries that make estrogen progesterone. There's some data that the astrocytes do that. Did you look at like estrogen specific or progesterone specific clustering of epigenetic changes?
We did not, but I think that it would be really awesome to look at something like that and really see, really tease out some of the sex differences that we observed. Because again, I'm just kind of giving a broad hypothesis of what I think could be happening, but we didn't test anything specifically. But you make a really great point about looking at maybe clustering. That would be very, very interesting to look at. I would love to do something like that.
Yeah, and also to make a small point, many of us, the stroke researchers, we think that it is important to use older females in stroke research. So we are using older mice and rats. But however, I hear from several people that what we call menopause in humans is not the same in rodents. So rodents go into a continuous cycling of some sort, and so I really don't know whether the studies, the rodent studies in older females can be similar to older females in humans. And so the epigenetics certainly are species specific and certainly are sex specific. So we need to take all these results with a pinch of salt.
Right, I agree. But there's some data now, at least that our astrocytes are like the supportive cells of the brain. They also make some level of estrogen progesterone that is independent of the menstrual cycle. So, and I don't know if there's an organ specificity to that or not. In terms of the clustering signature, but since you're doing an epigenetic analysis, it might be interesting to see if the epigenetics of estrogen progesterone on the astrocytes is different than say on the sex organs.
Right.
That would be really interesting.
Kahlilia, this is Gurvathan calling. I wonder, you have chosen this IP injections of ascorbate. What do you think about IV injections?
Yes, so that's a really great question because we know with IP that you have kind of slower delivery than you would have with IV. And so we didn't do IV, but I think that that would be kind of the next step in kind of looking at modes of delivery and testing the therapeutic effectiveness, because that would most likely be the mode of delivery that would be used in clinic, an IV injection. And that's what's used right now in clinical trials of ascorbate for sepsis, for example. They do IV injections. So I think looking at IV would also be really great. And we could see if, with IV injection, would it be even more effective than what we see with IP?
Thank you.
Thanks, Kahlilia. I think most of the clinicians has to leave pretty soon, or already left, and so thank you for giving this talk so early in the morning today, and we all learned a bit of what you are doing and about epigenetics, and good luck in your research.
Okay,
Yeah, thanks
great. Thank you
everyone.
very
Bye.
much.